we are developing a miniaturized eye model known as the eye-on-a-chip device to facilitate the screening and testing of new drugs and products for the eye. This device will help determine whether a new drug or formulation is safe and effective. To create this model, we leverage technologies such as microfluidics and cell cultures, and have developed new image analysis to evaluate how cells respond to various drugs and products.
As scientists, we are constantly developing new products, new formulations, and treatments for the eye. What we lack are reliable models to assess their safety and efficacy, especially in the early stages, because without it, it's really hard to know if we are on the right track. There are in vitro models that we can use, but they are often very simplistic and do not reflect the environment of the human eye. Therefore, a lot of the results that we do obtain in vitro are often not reflective of what you would see in human trials, and that is a big problem.
It starts not only with an idea, but with a problem. Researchers like us and even big pharmaceutical companies are constantly developing new eye treatments, but we all face the same issue. There is not a reliable way to test how safe or effectiveness these products are. At the same time, we are not yet at the stage for animal studies or clinical trial. Consequently, there is a clear need to develop a model that can bridge this critical gap. We began by listing the essential features of a model needed: high throughput, minimal sample and test solution requirement, the ability to replicate key parameters of the eye, such as low flow rates, and compatibility with traditional cell assays, including microscopy. From there, we explored how technologies like 3D printing, microfluidics, and cell culture techniques could help us achieve these goals.
We are perhaps the greatest beneficiaries of our own invention, as we can use it to test our own products. Researchers can also use our models instead of developing their own, and can instead use those resources toward their product development. On a broader scale, the research community and society as a whole will benefit from more efficient use of R&D resources and a faster pipeline for bringing new products to market. Early stage testing helps vet products more effectively, reduce wasted time and money later in clinical trials, while increasing the likelihood of successful commercialization.
We have a strong understanding of the eye and cell, but building an advanced eye-on-a-chip model requires expertise from other fields. Fortunately, one of our key advantages is being part of the Center for Eye and Vision Research, CEVR, which we have leveraged to connect with experts who contribute to this project. Without their help, we won't be able to develop our technologies. Beyond that, our connections with CEVR also provide access to seed funding opportunity, allowing us to develop our initial prototypes. We used the prototype we developed to test our own products, making us the first user of our own invention. In addition, we reached out to our colleagues for pilot testing. Most importantly, we engaged not only with other researchers early on, but also with industry professionals and companies that could potentially utilize our technologies. Overall, the feedback we received was positive, and we were able to identify the key features needed for the final product.
We are still in the early stage of our invention, with much more work ahead. Developing a prototype cell-on-a-chip model is just the first step. The real challenge will be how to scale production to thousands of these biological models while ensuring consistency and minimizing variability. Our future focus is on making these eye-on-a-chip models robust, reliable, and easy to use. Achieving this will require a much deeper understanding of cellular biology that goes beyond our in-house expertise, making collaborations with new experts essential. Additionally, we are also trying to develop new assays, such as automated image assays, to enhance and leverage our models for new, insightful drug screening.
Don't get caught up in ideas because everyone has them. What truly matters is finding a problem worth solving, one that impacts a lot of people, ideally including yourself, so that solving it feels meaningful and valuable. Second, you want to ensure it's a problem that you can solve, either through your own expertise or by leveraging your connections with experts. The sweet spot is really where a significant problem intersects with your ability to solve it. And lastly, you want to be able to convince others to join you, whether by contributing their expertise, offering support, or providing funding to help turn your solution into a reality. And I will leave this quote here. Alone you can go fast, but if you want to go far, go with a good team.